Hi everybody, it's Mr. Matthew here again for the MCAS video series on heredity, inheritance, and variation in traits. And this is video 4 in our series. In this video, we're going to talk about some specific information to illustrate how genetic traits of individuals and the presence of specific alleles in a population are due to interactions between the genetic factors and environmental factors. So we're going to look at a few specific traits and how those traits vary a little bit in different location, and we'll also talk about how environmental factors may activate or turn on or modify certain genes within an individual's lifetime. So all of these things are going to come together to see how populations might be impacted by the environment and how that environment plays a role in which phenotypes we see expressed in given locations. So here we go. So our first example that we're going to talk about over here on the left, and that's looking at blood type. And we went through blood type pretty extensively in video number 3, but this is a good example of looking at multiple alleles for one gene. And so the reason that you're going to see variation within blood typing is because there are more than just the dominant and recessive allele present in blood typing. As a result of this multiple allele, you actually have 4 different phenotypes, and from varying crosses, you're going to get greater variation. So this is one example of a factor that will influence the trait of individuals. If instead of just being simply a dominant recessive trait, you're going to have more factors that's going to complicate the inheritance pattern. Another example we could talk about is what happens if you have multiple genes involved. So in this particular instance, we're looking at the ABO blood typing, but you've probably also heard about the idea that blood typing has a positive and negative. So in that instance, what we're going to be looking at is what's known as Rh factor. And so Rh factor is another gene that influences blood type. And so in addition to the fact that you might have type A blood, you could have a separate gene that would dictate whether or not you have a different protein, an Rh protein present or not. As a result, rather than just seeing, 1 or 2 predominant phenotypes, you end up seeing a range of phenotypes in the population, and those graphs tend to look more like a bell curve, and you end up seeing broader variation within the population. So that's one factor that influences the distribution of traits. Another type of factor that you might have is what happens if you have a trait that has more than one phenotype. And so again, we looked over here on the right as our example of malaria and sickle cell anemia. And this is a good example of a trait in the case of sickle cell trait where there's more than one factor that occurs as a result of sickle cell trait. Now, we focused in on here, on this distribution here, that when you have malaria present, you end up seeing an increase in sickle cell trait, and that provides malarial resistance. But another factor that we also see is that you end up seeing an increase in sickle cell disease. These are 2 completely different phenotypes that are at play, and they end up having an influence on one another. So in this case, it's a single gene and having multiple phenotypes. This is sort of the opposite of what we saw with blood type. This is one particular gene, and it's having multiple different phenotypes as a result. All right, so another factor to keep in mind when we look at these factors is the idea that there's an interaction between genes and the environment. And so for this, what we're looking at is that individuals are born with a predisposition to have certain phenotypes in their lifetime. But just because you're predisposed to have a phenotype doesn't mean that you're actually going to express that specific phenotype. Certain diseases like heart disease or cancer, you may have a certain genetic predisposition to developing these conditions. But the environment that you are in will play another role. And so over here on the right, I have all these different risk factors that we see for heart disease. So let's say you are born with a genetic predisposition to develop heart disease. Well, if you end up taking in a diet that's very high in cholesterol, you're going to dramatically increase your risk of developing heart disease. Similarly, if you abstain from cardiovascular exercise, you will increase your risk for heart disease. If you smoke, if you don't maintain a healthy weight, if you do factors that influence your blood pressure, all of these things will play a factor on whether or not you would ultimately develop heart disease. And so the important thing here is that genes are not necessarily destiny. And a lot of times when we talk about genes and disease, we talk about things that are very strongly linked. So for example, I talked about in our last video, color blindness and the phenotypes that you get as a result. Now, if you have the color blindness alleles and you're a male and you have that one color blindness alleles, you will have a hard time. You will not be able to discern between reds and greens. But more often than not, we have these polygenic traits that produce risk factors for specific traits. And for those cases, you have to combine both the environment and your lifestyle along with your genetic predisposition to determine if you're actually going to have certain genes turned on and turned off. It would be nice if there was a simple formula where you'd say, if you eat this, you get this, or if you do this, you do that. But the fact is that it's a much more complicated and much more nuanced, and it's also much less direct. It's not like you'd say, all right, if you have 5 cheeseburgers, you're going to be okay and not get heart disease. But if you have 10 cheeseburgers, you're going to be in trouble. It's a much more nuanced factor. And as a result, you have some really messy data when it comes to genetic risk factors. We can give you some broad categories about health, but every individual is going to be a little bit different, and how your body interacts with those risk factors will also vary. We are approaching the point where we will get more specificity about personalized medicine and personalized genetic predispositions in the next decade or so. I expect those things to become much clearer, but whether or not everyone chooses to have those genetic screens done will be also determined largely on money. It's going to cost money to run those genetic tests, and not everyone will have access to those things. All right, so back to our malaria sickle cell disease example. And now what we're going to do is talk about the maintenance of alleles for sickle cell anemia and its high frequency related to malaria in certain regions because of that resistance. So I just want to come back to this idea that in addition to our underlying genes and our lifestyle or the environments that we impose on there are outside factors that do play a role. And one classic example of that is this malaria example that we should keep coming back to over and over again. And the fact is we often don't think about humans as being influenced by natural selection. We control our environment in a very, very profound way. But we can see in this region of Central Africa where the environment and the presence of that malarial parasite is having a huge impact on allele frequency. And in the parts of the world where you do not see endemic malaria, you do not end up seeing high incidence of sickle cell disease. Having a disease or having a predisposition to developing issues with your red blood cells, even if you're heterozygous and you only have mild factors, that puts you at risk and you are not going to be as fit as those without the trait. But if you put humans in an environment where having those red blood cells that are altered provides a fitness advantage and they're better able to survive and reproduce. Specifically, children are at great risk. And so if you have these sickle cell traits and you're allowed to survive because of that presence of that trait and survive infancy and childhood and reach to a reproductive age, you're going to pass those traits on. So even though we don't think of humans as being strongly influenced by natural selection, this is a good example of how humans are under the forces of natural selection in a very clear and obvious way. All right, so hopefully at this point you've got a pretty good handle on the fact that there are different things that influence the traits of individuals. The types of alleles that will occur in a population are different factors that will lead to allele distributions within a population, and also how your genes interact with environment, ultimately leading to your fitness and the phenotypes you express. A couple of examples we talked about are multiple alleles with blood typing and how multiple genes will influence a trait like that of tissue type. And then you should also be aware that lifestyle, in our case, our diet, is another example of environmental risk. We could have very easily gone down the line and talked about how exposure to UV light plays a role in both skin cancer and skin color. And there's a lot of other examples that are there. We may talk a little bit about the 5 factors that are associated with maintaining what we call the Hardy-Weinberg equilibrium, but you will not have to do any calculations of Hardy-Weinberg for the state MCAS test. All right, so I hope that was helpful to everyone, and I'll talk to you soon.